I don't have a very long introduction for Cody. Cody came to University of Chicago, was initially interested in MD, and I don't know what happened. He decided to take the PhD at the same time, right? So that's good for us. And he joined my lab. I have to say, at the beginning, both he and I were not sure what we should work on. We messed around on several projects, and eventually Cody realized that he should not rely on me. So he went out and figured out his own thesis direction, his own thesis projects. So basically what you're going to hear today, pretty much his own idea. All I didn't do much compared to Tao, Marcus, Aaron. We'll just be cheerleaders on the side in the last several years. I think the committee members will agree with me. So Cody, it's your show.
Great. Let me share my screen. Okay, can you guys all see my screen?
Yes.
Okay. So thanks for that kind introduction, Chuan. Today I'm pleased to present my thesis entitled Determinants of the m6A Epitranscriptome. Gene expression refers to the process by which genetic information flows from the 4-letter nucleotide alphabet of DNA and RNA to the 20-letter amino acid alphabet of protein. Genes encoded within DNA are transcribed into a complementary sequence of pre-mRNA, which then undergoes several RNA processing steps, including capping in the 5' end, splicing to join the exons and remove the introns, and polyadenylation of the 3' end. This mature mRNA is then translated by ribosomes into protein. Proteins are the principal workhorses of the cell. They allow a cell to generate energy, to grow and divide, and to perform specialized functions. Notably, the rate of gene expression determines the amount of any particular protein within a cell. Therefore, control of gene expression is a principal determinant of cellular form and function. So the 4 basic building blocks of RNA are A, U, C, and G, which are the nucleosides adenosine, uridine, cytidine, and guanosine. Notably, there are also over 150 non-canonical modified nucleosides in RNA. These non-canonical nucleosides consist of modified forms of the canonical nucleosides, including acetylation, methylation, and isomerization of the base, as well as methylation of the ribose sugar, among many others. Collectively, these RNA modifications are termed the epitranscriptome because they comprise a layer of chemical information on top of the primary RNA sequence. The structures I've depicted here are characterized mammalian mRNA modifications, as that will be the focus of my talk today. So how are mRNA modifications installed and what do they do? So mRNA modifications are installed by cellular enzymatic machineries. These modifications alter the properties of mRNA. For example, many mRNA modifications have been reported to affect gene expression by changing the way that the mRNA interacts with the gene expression machinery. RNA modifications can not only be found in naturally occurring mRNAs, but also they can be incorporated into synthetic mRNAs for translational applications. So here's a picture of me getting the Pfizer mRNA COVID-19 vaccine, which many of you may have also received. And what's interesting is if you look at the sequence, you can see that all the U's in the sequence have been replaced with Psi's. And in this context, Psi corresponds to the RNA modification N1-methylpseudouridine. And the reason N1-methylpseudouridine is incorporated into this RNA is because it increases mRNA stability and decreases immunogenicity, therefore increasing the efficacy of the vaccine. So altogether, RNA modifications are important regulators of mRNA function. In my talk today, I will focus on m6A, which is the most prevalent mRNA modification in mammalian mRNA. m6A consists of adenosine methylated at the N6 position by the metal-3-metal-14 RNA methyltransferase complex. There are tens of thousands of m6A sites across the transcriptome, and metal-3-metal-14 deposits m6A within a DRACH consensus sequence. Notably, m6A deposition is highly specific. Only a small fraction of DRACH sites are actually methylated, and a subset of genes contain m6A in their transcripts. In transcripts that contain m6A sites are strongly enriched within long internal exons and near-stop codons. m6A plays a variety of crucial roles in a diverse array of biological processes. So I don't have time to go into each of these individually today, but broadly speaking, m6A has been shown to be important for many developmental processes that involve the differentiation of progenitor cells into more specialized cell types. It's been shown to be important in the immune system through facilitating a variety of immune cell functions. And additionally, m6A has been implicated in many diseases, including many types of cancer, viral infection, and type 2 diabetes. Today, I'll just spotlight the role of m6A in acute myeloid leukemia. It's previously been shown that in AML, m6A is important for the initiation and maintenance of AML. Notably, it's been recently reported that a metal 3 metal 14 catalytic inhibitor, when given to mice given a preclinical model of AML, it prolongs the survival of the mice and reduces AML progression. So because of the importance of m6A in this disease system, it appears that targeting m6A methylation may have therapeutic potential. So the reason why m6A is so important in many diverse biological processes is because m6A plays multifaceted roles in gene expression. m6A is deposited onto mRNA co-transcriptionally, and upon deposition, it can be recognized by various m6A reader proteins. One family of m6A reader proteins includes the YTH family, which contains a specific domain that can specifically recognize the m6A moiety. Other proteins that preferentially bind m6A include HNR and PC and HNR and PG, which do not directly recognize the m6A mark. However, the m6A residue alters the secondary structure surrounding the m6A site such that these proteins can bind to newly accessible RNA sites. So these diverse reader proteins have varied effects on downstream gene expression. They have been reported to regulate alternative pre-mRNA processing, nuclear export, mRNA stability, as well as translation. Notably, specificity of m6A deposition is central to m6A-mediated gene regulation. So first, specificity in terms of methylation of transcripts from a specific subset of genes is important because methylation of specific transcripts allows these transcripts to be regulated by this m6A pathway that I previously described, whereas transcripts that are not methylated can escape this regulation. Further, there's evidence to suggest that methylation of specific sites within a transcript may also be important because the function of m6A on mRNA appears to be context specific. So using translation as an example, m6A in the 5' UTR can promote cap-independent translation initiation by recruiting the translation initiation factor EIF3. m6A within the coding sequence can regulate translational efficiency through direct interactions with the ribosome. And m6A in the 3' UTR can recruit the reader protein YTHDF1 to promote translation initiation. So due to the importance of m6A specificity in m6A-mediated gene regulation, how specificity of the m6A epitranscriptome is controlled is one of the biggest questions in the field. So when thinking theoretically about the important determinants of m6A specificity, you can break them into two broad groups. So one is intrinsic determinants. And what I mean by intrinsic determinants is I mean factors that alter the intrinsic efficiency with which the methyltransferase complex can deposit m6A on a particular RNA sequence. Examples of this would include the local RNA sequence and secondary structure. Additionally, there's another category that I'll call extrinsic determinants. And what I mean by this is I mean external factors that can regulate the ability of the methyltransferase to deposit m6A on specific sequences. So previously, it's been reported that transcription factors, RNA binding proteins, histone modifications, and RNA polymerase can interact with the methyltransferase complex and promote deposition of m6A at specific sequences. So notably, the relative importance between intrinsic and extrinsic determinants is not very well understood. And this is important because it has implications for the nature of m6A as a regulatory system. You can imagine that if intrinsic determinants dominate, then this would imply that m6A may be largely hard-coded within the RNA sequence and therefore paints a fairly static picture of m6A. In contrast, if extrinsic determinants are predominant, you can imagine that if the cellular state changes and the levels or activities of these various factors that regulate methylation vary, that you would get changes in m6A, and this would paint a much more dynamic picture of m6A methylation as a regulatory system. Additionally, known mechanisms that I've described up here do not appear to explain the enrichment of m6A at specific transcriptome features globally, as well as much of the selective deposition of m6A on specific mRNAs. Therefore, we hypothesized that there are unknown mechanisms that have not currently been discovered that control global m6A specificity. So the question that we wanted to answer was, what determines which mRNAs and which mRNA sites are m6A methylated? So first, what is the relative importance of intrinsic versus extrinsic determinants in controlling m6A specificity? And second, what are the unknown mechanisms that control the global specificity of the m6A epitranscriptome? We hypothesized that addressing the first question would allow us to better address the second. So we started with the first question, and we developed an assay that I'll call massively parallel assay for m6A. And the question we can answer with this assay is very simple. Is local sequence sufficient to specify the endogenous methylation status of an m6A site? And with this assay, we're able to ask this question in a very high throughput manner at thousands of sequences simultaneously. So to take you through the workflow of this assay, we know where the endogenous locations of m6A sites, or conversely unmethylated DRACH sites, are in the transcriptome. So what we did was we synthesized the local sequence surrounding these sites. So around about 100 nucleotides surrounding either the methylated or unmethylated sites. We clone these into a plasmid library and then express them. And essentially what this does is it uncouples the local sequence from its larger endogenous context. And so what we did next was we precipitated this expressed RNA with an antibody that recognizes m6A. And then we perform next generation sequencing to assess the methylation status of each of these sequences. And the fundamental question we're asking here is, how does the endogenous methylation status compare to the MPM6A methylation status? So just to take you through an example, for this blue sequence here, it's methylated endogenously, and it's also methylated in the artificial context of this reporter. So local sequence appears to be sufficient for this sequence. Conversely, for the orange sequence, it loses methylation when you express it in the context of the reporter, and therefore local sequence is not sufficient. Finally, for this purple sequence, it actually gains methylation when you express it in the artificial context, and thus local sequence is again not sufficient. So we performed this assay with about 10,000 endogenously methylated and unmethylated sequences to survey this on an epitranscriptome wide scale. And what we found was quite surprising to us. We found that endogenously methylated and unmethylated sequences are methylated to comparable levels in the MPM6A reporter. Here you can see the endogenously methylated group, and that the vast majority of these sequences are significantly methylated in the MPM6A reporter. Interestingly, in the endogenously unmethylated group, also the vast majority of these sequences were significantly methylated in the MPM6A reporter. And if you look at their enrichment scores overall, you can see that they're quite comparable. To give you an idea what this looks like for individual sequences, here's an endogenously methylated sequence that is also methylated in the MPM6A reporter. And here are two endogenously unmethylated sequences that become methylated in the MPM6A reporter. And we observe this through transfection into cells as well as in vitro transcription and methylation. So what this suggested to us was that local sequence is insufficient to specify the correct endogenous methylation status of an m6A site, and that methylation is suppressed at thousands of potential m6A sites across the transcriptome. Next, we further investigated the characteristics of these suppressed m6A sites. When we visualized them on a metagene plot, what was interesting was we observed that they formed an inverse distribution to endogenously methylated sites. They are enriched within coding sequences preceding the stop codon, as well as in 3' UTRs following the stop codon. Additionally, we found that they were enriched in much shorter internal exons than the typical endogenous M6A site. And these much shorter internal exons are actually more in line with what the average internal exon length is in the transcriptome. Finally, we found that a significant proportion of these suppressed sites reside within transcripts that are usually completely unmethylated. So overall, our MPM6A results suggest that suppression of M6A in unmethylated transcriptome regions is a major determinant of global M6A specificity. So going back to this picture of how M6A specificity is controlled, as I previously mentioned, most previously reported mechanisms involve promotion of methylation at specific regions by various transacting factors. However, based on our results from the m6A assay, we realized that we might need to flip this question on its head in order to understand m6A epitranscriptome specificity. And that a relevant question would be, what is suppressing methylation at mRNA regions that are normally not methylated? And that this could be an important contributor to global m6A specificity. So no known mechanisms appear to account for this suppression. So next we asked, what are the mechanisms that suppress methylation at these sites? So the first hint that we got about what might be happening here is we found that spliceosome components are enriched at suppressed m6A sites. And this makes sense if you think back to our earlier finding that suppressed m6A sites are enriched in much shorter internal exons than endogenous m6A sites. And this is because m6A sites within shorter internal exons would, by definition, be much more likely to be closer to the nearest splice site and the spliceosome and the splicing machinery. So this led us to ask the question, does residence of a potential m6A site in an average length internal exon and proximal to splice sites suppress m6A deposition? So the strategy that we took to assess this was we took a suppressed m6A site derived from an average length internal exon that we identified by MPM6A. And what we did was we either cloned it into a mini gene reporter that contains a first internal last exon, with the internal exon being about average length, or into an equivalent reporter with the introns removed, such that the mature mRNA sequence is the same, but the only difference is that one is spliced and the other isn't. Sorry, I'm getting some feedback. If someone's not muted, could you please mute yourself? Thank you. And what we found was that on the spliced construct, consistent with our MPM6A results, you could see that M6A this suppressed M6A site becomes activated. But conversely, in the context of the spliced construct in the internal exon, you can see that there's actually strong suppression of this M6A site. And further, if you expand out the length of the internal exon by cloning in more and more flanking exonic sequence surrounding this suppressed M6A site, you can see that you gradually lose this ability of, this suppression that we observe in the smallest internal exon. And you can see that once you get past around 400 nucleotides, you actually lose this suppression. And if you compare the length scales here to the distribution of internal exon length throughout the transcriptome, you can see that 90% of internal exons are actually less than 250 nucleotides. So this mechanism could potentially suppress thousands of M6A sites within internal exons encoding sequences across the transcriptome. So next, we wanted to identify the specific cellular factors that were mediating the splicing-dependent, exon length-dependent suppression of M6A. And so one candidate factor we considered was the exon junction complex, or EJC. The EJC is deposited onto mRNA by spliceosomes 24 nucleotides upstream of exon junctions. And so the core factors of the EJC are MAGO, RBM8A, and EIF4A3. And upon deposition onto the RNA, it associates with peripheral factors and can control nuclear export, nonsense-mediated decay, translation, as well as mRNA localization. In addition to these more established roles, it's recently been reported that in two recent studies that the EJC can suppress local cryptic splice sites. So it appears that the EJC forms a zone of protection around proximal RNA regions that protects the RNA from splicing. So we hypothesized that the EJC could be mediating the splicing and exon length dependent suppression that we previously observed. And the reason for this is because if you consider suppressed M6A sites that reside within average length internal exons, they would be in relatively close proximity to both an upstream and a downstream EJC. In contrast, methylation within long internal exons and near stop codons could be hundreds of nucleotides removed from the nearest EJC. And for stop codons, the reason why this is because stop codons generally reside within the beginning of last exons, which is downstream of the last exon junction. Additionally, EJCs could potentially completely suppress methylation on transcripts because most transcripts do not contain long internal exons. And therefore, this mechanism and this hypothesis appear to nicely fit with the known characteristics of m6A specificity. So next, we wanted to test whether this really appealing hypothesis was true. And what we did was we in human cell lines, we depleted EIF4A3 and RBM8A, which are two core EJC factors. And what we observed was that there was global upregulation of m6A on mRNA when you deplete these factors. And notably, we see a stronger effect when we deplete EIF4A3, which could be due to the fact that we have stronger knockdown efficiency of this factor. So looking transcriptome-wide using m6A sequencing technology, we find that thousands of mRNA regions are hypermethylated upon depletion of the EJC core factors. And the regions in red correspond to regions that are actually not methylated at all in the control condition. So these are newly methylated transcriptome regions. So to give you an idea of what this looks like on specific genes, here is a gene SRSF6. You can see in the wild type condition that there is methylation in the first exon as well as near the stop codon in the last exon, but not much in the internal exons in the coding sequence. However, upon EJC depletion, you can see that there is strong hypermethylation of internal exons of average length within the coding sequence. Furthermore, you can also observe this on transcripts that are actually not methylated at all in endogenous conditions. And you can see that when you deplete the EJC, you get strong hypermethylation of these internal exons of around average length. So looking at this more systematically, we observed that hypermethylated regions are enriched in internal exons of around average length transcriptome wide, and that actually when you deplete EIF4A3, you lose the specificity of m6A for long internal exons due to the de-repression of these m6A in much shorter internal exons. So overall, the EJC appears to mediate enrichment of m6A in long internal exons. So looking at exon junction proximal regions and looking at the m6A levels there, this result also supported our initial hypothesis because you can see that there's a global elevation of m6A levels upon EJC depletion. Furthermore, I'll note that it's previously been reported that the m6A stop codon enrichment can be more precisely described as a peak of enrichment that begins 150 nucleotides downstream the start of last exons. And what you can see is that when you deplete the EJC, you actually get an increase in m6A enrichment in this last exon start proximal region. So overall, it appears that the EJC also mediates the rise in m6A leading up to the stop codon that is alternatively visualized as this rise following the start of the last exon. And finally, if you look at the hypermethylated regions on a metagene plot, you can see consistent with the fact that internal exons generally reside within coding sequence, that the coding sequences, that the hypermethylated regions generally reside within coding sequences. And again, you can see that the global distribution of m6A is shifted with much more m6A density in the region preceding the stop codon in the coding sequence. Additionally, while most of these hypermethylated regions appear on transcripts that have at least one m6A site under normal conditions, we also find that over 1,000 of these transcripts don't contain any m6A sites in normal conditions. And therefore, the EJC also contributes to the restriction of m6A to a specific subset of transcripts in the transcriptome. So overall, this data indicates that EJC suppression of m6A is a major mechanistic determinant of methylation that mediates multiple aspects of global m6A specificity. So the next question we wanted to ask is, what functional consequence does EJC suppression of m6A have on gene expression? So the strategy we took to address this question was we depleted EIF4A3 in both wild type cells as well as metal-3 heterozygous knockout cells that are deficient in m6A. And in gene, I'm depicting genes for which the gene expression change upon EIF4A3 depletion is more positive in the m6A deficient cells than in the wild type cells. And in blue, I'm depicting genes for which the change in gene expression upon EIF4A3 depletion is more negative in m6A deficient cells compared to wild type cells. And overall, we identify hundreds of hypermethylated transcripts for whom m6A appears to contribute to the upregulation or downregulation of these transcripts upon EJC depletion. To highlight a specific example, I'll focus on TP53, which is the gene that encodes p53. And so the reason why we focused on this particular gene was because it's previously been reported that EJC deficiency in mouse neural stem cells causes p53-mediated microcephaly. And this may be a relevant model for human neurodevelopmental disorders that are associated with EJC mutations. So these authors showed that EIF4A3 mutants show significant upregulation of P53 in these mutants, and that this mediates the microcephaly phenotype. However, the mechanism behind how EJC depletion leads to P53 upregulation was not clear. Interestingly, in our cell lines, we observed that P53 mRNA is aberrantly methylated upon EJC depletion, and that at both the RNA as well as the protein levels, P53 appears to be upregulated upon EIF4A3 depletion in an m6A dependent manner. So you can see here in wild type cells that there's robust upregulation of P53, similar upon EIF4A3 depletion, similar to what's observed in mice. But you can see that in m6A deficient cells, this upregulation is greatly diminished. So altogether, this data indicates that suppression of m6A appears to be a previously unrecognized mechanism by which the EJC controls gene expression. So next, we've seen that we can robustly observe the importance of EJC suppression of m6A specificity in the cell lines that we've been working with in vitro. And next, we wanted to assess the extent to which this mechanism may be important in vivo in human tissues, because ultimately that's what we're trying to model with our cell lines. And so the approach that we took to assess this was we took the EJC suppressed regions that we identified in our cell lines, and we asked, are these EJC suppressed regions also not methylated in 25 different human tissues for which m6A transcriptome wide mapping data is available? And what we saw was that the vast majority of these EJC suppressed regions are also not methylated in tissues. And this suggests that this mechanism is widely active in vivo and is important for m6A specificity in this context. And this actually was not very surprising to us, because it's known that the global features of m6A epitranscriptome specificity, such as the enrichment in long internal exons and near stop codons, is broadly conserved across cell lines and tissues. Interestingly, however, we noted that there were a small subset of EJC suppressed regions that appear to escape suppression in some tissues, but not others. And these collectively comprised about several hundred different EJC suppressed regions. And so the next question we wanted to ask was, what are the mechanisms that allow these regions to escape EJC suppression in certain tissues? And the reason why we were interested in this question was because not much is really known about the mechanisms that control m6A variation across different tissues or across different physiological contexts in general. So we reasoned that by understanding these mechanisms of how these regions are escaping EJC suppression, it may allow us to learn more about how m6A varies across different physiological contexts. And so we found that for a subset of these regions, it appears that methylation of alternative transcript isoforms with long exons mediates the escape from EJC suppression. So here are two regions that I'm highlighting that are representative of about 100 or so regions that we believe may be regulated by this sort of escape mechanism. And you can see that these two internal exons of around average length contain EJC suppressed m6A sites. And you can see that in certain tissues, these regions escape methylation, and that these m6A peaks actually span the canonical exon-intron boundary of the RNA. And so these appear to correspond to non-canonical alternative transcript isoforms that don't use this proximal splice site. So in the case of BAM120A, the methylation here appears to be driven by an alternative polyadenylation event within the intron. And for SGSM2, this appears to be driven by a retained intron. And you can see that these alternative transcript isoforms essentially convert what was an average length internal exon to a longer composite terminal exon or a longer composite internal exon with displacement of the downstream EJC. And so to assess whether this escape mechanism appears to be relevant quantitatively, what we did was we compared the exon-intron junction m6A read coverage that corresponds to these non-canonical transcript variants to the m6A enrichment across different tissues. And you can see that the exon-intron junction read coverage appears to explain a significant amount of variation in m6A across tissues. And further, we have some experimental evidence to support this model as well. So going back to the mini gene reporter that I discussed previously, we realized that if we remove either the first intron or the second intron and essentially convert the internal axon into a longer terminal axon, that we were able to, that this removal of an upstream or downstream exon junction is sufficient to partially de-repress a suppressed m6A site within an average length internal axon. And so these perturbations in gene structure are similar to what physiologically occurs in these alternative pre-mRNA processing events. So altogether, this data indicates that exon architecture is an important determinant of m6A methylation. And so further evidence supporting the importance of exon architecture in m6A methylation is the fact that we observed that mRNA produced from unspliced expression constructs is hypermethylated. So here are two genes for which we identified contain EJC suppressed m6A sites. And notably, these two genes are actually used in gene therapies for the treatment of monogenic disorders. And in this context, they are expressed from expression constructs that usually do not contain endogenous exon architecture. And so what we would predict based on our MPM6A and mini gene results is that expression of these genes from constructs that lack endogenous exon architecture may cause hypermethylation of this mRNA. And indeed, that is what we observe. So altogether, this supports the model that endogenous exon architecture and consequently endogenous splicing and exon junction complex deposition enable suppression of m6A methylation, while a lack of this endogenous exon architecture can cause hypermethylation of mRNA. So finally, we wanted to ask the question, how do EJCs mechanistically protect proximal RNA from m6A methylation? And so it's previously been noted that EJCs package and compact mRNA into messenger ribonucleoprotein particles, or mRNPs, and can protect long stretches of proximal mRNA from in vitro nuclease accessibility. So in this paper from quite a few years ago now, if you pull down EJCs and you digest away accessible RNA with nuclease, you can, and then you visualize the size of the distribution of the RNAs, you observe short footprints that you would expect based on the standard RNA binding protein binding to a particular RNA sequence. But in addition to these shorter footprints, you also observe an abundance of longer footprints that can reach up to 150 nucleotides in length. And so the authors of this study proposed that the EJCs perform a packaging function in which they multimerize with other EJCs and associate with various other factors to package and compact mRNAs into mRNPs. And this explains how these exon junction complex proximal long footprints can be protected from nuclease accessibility. So this protection of long stretches of RNA from nucleus accessibility seemed to us to be reminiscent of what we were observing with m6A, where you can observe that EJC suppress long proximal stretches from m6A methylation. So next we asked whether this EJC packaging function was in any way relevant for what we were observing for m6A suppression. So we isolated these EJC protective footprints, and consistent with what was reported in the literature, you can see that we observed long RNA footprints. And interestingly, if you look at the m6A levels on these long RNA footprints, you can see that they are strongly depleted of m6A. And what this suggests is that not only can EJC packaging protect proximal RNA from nuclease accessibility, but in the cell, it can protect these long stretches from RNA methylation. And further, when we took these EJC-bound RNA footprints and we subjected them to an in vitro methylation reaction with a recombinant metal 3 metal 14, you can see that these RNA footprints also seem to be protected from methylation. In contrast, when we spiked in an unmethylated RNA that is just free and not bound by the EJC, we observed robust methylation. Furthermore, the lack of increase in methylation here, we checked to make sure that this wasn't just because there weren't any m6A sites available for methylation on these sequences, because if you take deproteinized RNA footprints and subject them to the same in vitro methylation reaction conditions that we use over here, you can see that there's robust methylation. So altogether, this suggests that EJC mRNP packaging blocks accessibility of local RNA to methylation. So next we wondered if EJCs sterically hinder accessibility of proximal RNA to methylation and nucleus digestion, is accessibility also hindered for other RNA regulatory machineries? Because you can imagine that if the EJCs are blocking methylation and nucleus accessibility through this general steric hindrance sort of accessibility mechanism, you wouldn't necessarily think that this would be specific for methylation or nucleus digestion, and that this could potentially apply to a broad array of regulatory processes on RNA. So as I previously discussed, it's been recently discovered that the EJC can suppress splicing in a mechanism that's reminiscent to what we find for m6A. And so what's interesting is if we compare the locations of the splice site, the suppressed splice sites, and the EJC suppressed m6A sites, we found significant overlap between these regions. And here I should say that the brackets of the ends of the brackets refer to the splice junctions that are suppressed by the EJC. And if you look transcriptome wide, you can see that globally, suppressed m6A sites co-localize with suppressed splice sites. And altogether, this suggests that exon architecture and EJC-mediated mRNA packaging may broadly determine local accessibility of mRNA regions to RNA regulatory machineries. And so altogether, this is a model that we've put together based on the data that I've described to you today. So EJCs appear to package local mRNA and suppress methylation deposition. This but however, regions that are distal to EJCs, such as in long internal exons and near stop codons, are accessible for methylation by the methyltransferase. If you deplete the EJCs, you not only lose regional m6A specificity, but you also lose transcript level m6A specificity, because EJCs can also completely suppress methylation on unmethylated transcripts. And we also propose that exon architecture and EJCs may broadly determine local mRNA accessibility to RNA modifying machineries, and that EJCs appear to suppress m6A methylation, splicing, and nucleus digestion. And we think it'd be interesting to investigate in the future whether additional regulatory factors may be impacted by this mRNA packaging function. So to summarize, in this presentation, I've described the development of MPM6A, which enables high throughput assessment of m6A methylation status of thousands of design sequences. We discovered that widespread suppression of m6A methylation is a major determinant of global m6A specificity. We identified EJCs as m6A suppressors that protect exon junction proximal regions from methylation. We find that exon architecture is an important determinant of m6A methylation, and that mRNAs with altered exon structures can enable differential methylation. We find that EJC suppression of m6A is essential for proper control of gene expression. And finally, we find that exon architecture may broadly determine the local accessibility of mRNA regions to regulatory machineries. And I think this is a particularly interesting point in terms of future work, because it's been well established for DNA that chromatin accessibility and the packaging of DNA by histone proteins is an important determinant of gene expression. However, the functional relevance of mRNP accessibility or mRNA packaging has not really been as thoroughly explored. And so in addition to so there is some evidence to support the packaging of pre-mRNA by hnRNP proteins. And I've also introduced to you today the evidence that EJC packages mRNA. However, the functional consequences of these packaging activities have not been that thoroughly explored. And I think it'll be interesting to see in the future the extent to which mRNP accessibility and this packaging of mRNA could be, to what extent it could be an important determinant of gene expression. And I guess that remains to be seen. So now I'll take questions, and then after questions are over, I'll finish with some acknowledgements.
All right, thanks, Cody. Questions from the audience?
Hey, Cody. Great talk.
Hey.
You mentioned that regulation of mRNA methylation may also be associated with different cell states, like if, for example, if the cell is undergoing stress it might methylate different regions. Do you believe this is associated with differential exon structures, or do you think there's another mechanism at place for these kinds of things?
Yeah, so what I'll say is I think that the mechanism that I sort of described in terms of alternative splicing leading to differential methylation, I think theoretically it could mediate differential methylation in the sort of context that you're describing. I'll also say that we observed that for EJC suppressed regions, many of these regions actually seem to escape suppression without changes in exon structure. So I think that there are probably many different mechanisms that are at play here. And there are also probably EJC independent mechanisms as well. So I think that there are probably a lot of different possibilities. Yeah, thanks.
Other questions?
Hi, Cody, I have a question. So you mentioned that EJC are main determinants of m6A deposition. You also mentioned there's only around 5% of our motif is that's m6A. So I wonder whether you calculate the region covered by EJC, how many motif sites, and how many of them like methylated after depletion of EJC compared with other region, the CDAs with some other covered by EJC. What's the change of m6A after depletion of EJC?
Sorry, I think you kind of broke up at the end there. Can you repeat the last part of your question?
So yeah, I think it's you have a very good story about EJC regulate the m6A deposition, right? So for the mRNA for different region, for example, there's EJC covered region and the EJC didn't cover the region, right? So after EJC depletion, they compared the percentage of the m6A deposition in the conserved motif.
Okay, I see. Yeah, so actually we haven't done any single base resolution m6A mapping for EJC depletion. So I wouldn't be able to tell you the specific fraction of sites because we only have the regions from the conventional m6A sequencing.
Yeah, okay. Another question is, since the EJC play a important function, but what factors can regulate EJC itself?
Right. So so one factor that can regulate, for example, EJC deposition. So I should say that EJCs are deposited onto all spliced mRNAs. But I think like one thing that we highlighted in this presentation was that if you have alternative processing events that cause differences in exon length, that can regulate where EJCs are positioned. So that's like one way that EJC can be regulated. I guess I should also mention that it's thought that the vast majority of exon junctions are bound by EJCs, but by transcriptome-wide mapping, it appears that maybe like 20% of them or somewhere in that range may not have EJCs. And I don't think it's currently clear whether that's something to do with just the sequencing technology or whether that's a true biological signal, but it's possible that there may be some variation in EJC deposition that is caused by various factors that are unknown. Good question.
Yeah, okay, thank you.
Other questions from the audience?
Hey, Cody, really good talk. I just have a small question, which is, I think there were a couple other hits when you were looking for what regulates suppression of m6A methylation, like DDX24 and AQR. Do you know what those things do? And did you look into just the EJC or anything else?
Yeah, I believe DDX24 was a helicase. I actually didn't test those other factors that came up just because we kind of got this EJC story and that's what I followed. I will say another factor was like UPF1. And actually I tested UPF1. UPF1 is actually a factor that associates with the EJC to mediate nonsense mediated decay. In that case, UPF1 knockdown doesn't seem to do anything. And I think that's just because it's sort of a correlation thing where it's binding near EJCs, but it's not actually involved in sort of suppression. So yeah, I'm not sure whether those other factors are causal or anything, but it's something that we could test. Yeah.
All right, thanks.
Well, Cody, I actually have an interesting question. In lung cancer or some other cancer form, mRNA tends to use proximal poly-A denylation APA sites, meaning they're losing their 3' UTR. so the consensus from people study this was that they lose microRNA, right? Because the longer isoforms will have more microRNA. Been always thinking about the impact of M6A. What's your thought?
Yeah, so I'd say for proximal polyadenylation, I'm not sure that if you're referring to whether the EJC may be relevant for that, I don't necessarily think that's the case because even if you have a proximal polyadenylation, the nearest EJC is still quite a bit upstream from that area. So I don't think that necessarily the EJC would play a role in that, but as you know, and we've discussed before, there is an association in terms of preferential m6A methylation of proximal isoforms over distal isoforms, and therefore that, and by unknown factors, and that could be irrelevant to what you were saying in terms of cancers and mediating differential methylation.
All right, we have Professor Shengyang He.
Hey, Cody, nice talk. I have one or two questions depending on your answer. First one is the paradigm you're kind of defining of EJC for M6A. Does this apply to other type of mRNA non-methylation modification? You think this is more global than the M6A?
Yeah, that's a good question. We haven't actually looked at other mRNA modifications. What I will say is that in terms of what kinds of processes might be suppressed by the EJC, both splicing and m6A methylation, which are the processes that I've described as being suppressed, they both occur very quickly after transcription. And I think that I wonder if that might have something to do with the fact that they're both suppressed by the EJC, because it's known that over time, the EJC actually remodels in composition, becomes less packaged, and also EJCs are stripped from mRNAs following the first round of translation. So I think it's potentially other RNA modifications could be affected by the EJC, but I think that it may be dependent on how they're installed, like where and when in the cell they're installed. But I think that some people in the lab might be testing that right now, actually. So I guess it'll remain.
Oh, okay.
Yeah.
Yeah, I thought that would be an interesting project to look at it.
Mm-hmm.
I think you answered my follow-up question. So thank you, Cody.
Okay, thanks.
Great. Yeah, actually, yeah, that's a very good point on pseudouridine. You would think with the slow RNA part, the machinery is bigger, so it might be restricted by EJC. But for just the individual pass enzymes, probably less restriction. But it would be very interesting to look at these pseudouridines with different writers and see their distribution in terms of, I guess, relationship with exon lengths, right?
Yeah, definitely.
Not just the pseudo-U, but which writer installs the pseudo-U, right? That would be a great one for people in the lab to take a look.
Yeah, definitely.
Yeah, for two-parameter muscle, one would think it's mostly slow RNA based, so one would think it would be EJC regulated. We should also take a look.
Yeah, definitely.
Yeah. Other questions before we go to the closed door section?
Oh, I still have acknowledgements, Chuan.
You saw what?
I still need to do my acknowledgements. I left that for after the question.
Your acknowledgement. Okay, yeah. Please.
Okay. Okay, can you see my screen?
Yes.
Okay. So there are many people who I would like to thank for their contributions to the work described in this thesis. First, I would like to thank my PhD advisor, Chuan. Without his tremendous support and mentorship, this work would not have been possible. Starting from my earliest days in the lab, I felt that Chuan treated me as a partner in the research process and respected my ideas, which empowered me to take ownership of my work. Throughout the highs and lows of my thesis work, I found his support to be unwavering. He was always generous with his time, and his tireless work ethic and passion for his craft were inspiring. I am thankful for the lessons he has taught me, which have fostered my growth as a scientist and will undoubtedly serve me well in the future. I would like to thank all members of the He Lab I worked with, past and present, for their support throughout my years as a PhD student. I feel blessed to have been able to work in such a highly collegial environment. In particular, I want to thank Brian, who has generously given countless hours of his time over the years to discuss my research and give his thoughtful perspectives. I would also like to thank Scott and Ray too, who gave valuable input on many bioinformatic analyses. I also want to thank Hailing, Jambua, Phil, Jun, Kathy, Tong, Chili, and Huilong, who helped train me early on in my graduate years. I would also like to thank Alan and Caroline, my fellow MD PhD students in the lab, for their camaraderie. I also want to acknowledge people outside of our lab who provided valuable input and assisted me with various aspects of my research. John Staley, Amelia Jocelyn, Shuai Wang, the University of Chicago Genomics Facility, and the DNA Sequencing and Genotyping Facility. Next, I would like to thank my committee members, Marcus Clark, Aaron Adams, Menjie Chen, Marcelo Nobrega, and Tao Pan. All of you played an integral role in the completion of this thesis, with each of you making your own unique contributions. I could not have assembled a better group. Thank you to Nancy Schwartz and Lori Risner in the GDDTP MD PhD program, as well as the Committee on Immunology and the Pritzker School of Medicine for supporting my graduate work. I would also like to thank the many friends that have supported me during my PhD, who have filled my life with joy and meaning these past 4 years in Chicago. We've had a lot of fun and created some great memories, and I'm looking forward to creating many more. Next, I want to thank my mom and dad. You both are incredible scientists and even better parents. Growing up, your dinner table conversations captured my young scientific imagination and inspired me to try to make my own contributions to science one day. I'm grateful for your unconditional love and support and having you behind me, cheering me on every step of the way, no matter what path my life takes. Because of you, my dreams are limitless. Finally, I would like to thank my wife, Jessie, who brings joy to my life every day. You have been there every step of the way, commiserating with the lows and celebrating the highs. Thank you for being my partner in this journey and believing in me and supporting my dreams. You inspire me to be the best I can be. And thank you all for listening.
Great, Cody. Thank you for that wonderful presentation. Great. Okay, I think we should just go to the private session. I think we can just stay here and then just kick everybody off.
Oh yeah, wait, whatever way you want to do it.
It's probably better because it's hard to find the other private link.
Congrats, Cody.
Thanks, appreciate it,
Let's
dude.
just stay here and we can